Hello, and welcome to Breaking Down Biology blog Q&A session. Today, we're going to be discussing an article published in the journal Cell titled KDM6B Dependent Epigenetic Programming of Uterine Fibroblasts in Early Pregnancy Regulates Parturition Timing in Mice. I'm joined here today by first author Dr. Tara McIntyre and senior author Dr. Adrian Erlebacher.
Thanks for having us.
Good to be here.
Thinking back to the start of the project, what made you consider histone modifications as a possible mechanism for controlling the timing of birth?
Yeah, so Adrian first became interested in histone modifications many years ago while studying the decidua, which is the specialized stromal tissue that surrounds and supports the fetus. Back in 2012, he published that trimethyl H3K27 plays a really critical role in repressing key T-cell attracting chemokine genes, which really helped us explain how this tissue layer protects the fetus from being attacked by maternal immune cells. Follow-up to this work showed that the decidua also undergoes global loss of this repressive histone mark alongside sort of broad gene activation in late gestation. But whether this loss of the mark directly triggered parturition onset was unclear. So that open question really motivated us to ask which tissue layer really required these H3K27 demethylases called KDM6A or KDM6B. And as you know, to our surprise, it wasn't actually the decidua at all, but it was these endometrial fibroblasts that really required KDM6B. And then from there, we asked how and when KDM6B was involved.
Cool, thanks for sharing. As you were going through this project, what was the most surprising or unexpected result that you obtained?
Yeah, this is a hard question for me to answer, I think, because there were quite a few moments that were really surprising or unexpected. I think for me, the most exciting one was the erosion discovery. I very vividly remember we got the cut and run sequencing data back the day before Adrian and I both left for a Keystone conference. And I think we pretty much spent the entire conference scrolling IGV and looking at the peak, sharing screenshots of like these really amazing regions of trimethyl H3K27 erosion. And very often they were nearby genes that we were really excited about. And I think going into it, I really expected this sort of all or nothing pattern where it was going to be either the mark was there or it was gone. And so seeing this sort of beautiful erosion was just totally unexpected. It was just such a fun moment to, like, it really got our gears turning, I think, in terms of how to analyze it and what to do next and what it could mean.
So your study was done using mouse models. What do you hope this research might change about how we think about pregnancy or preterm birth in humans? Do we have any evidence that a similar mechanism of labor timing is operating in humans, especially given the fact that human pregnancy is much longer than a mouse pregnancy?
Yeah, that's a great question. And Tara and I talk about that all the time. Yeah, we don't know whether what we've discovered in mice is relevant to humans at all. That's the big question. And, hopefully it'll inspire people, either our lab or others, to look at human specimens or maybe non-human primates. These are obviously very difficult experiments, but I think the implications are potentially, if it turns out to have the same kind of pathway, are really important, because right now, people thinking about the pathogenesis of preterm labor, which is this really big clinical problem affecting, upwards of 10% of all pregnancies worldwide, major causes of neonatal morbidity and mortality, et cetera. People think about the pathogenesis of this with respect to events that are happening in late pregnancy. Clearly, if you have an infection, that can cause preterm labor, but many cases of preterm labor are not noticeably associated with at least clinical indications of infection. So nobody really knows what's going on with those cases. So what Tara's discovery that this timer is programmed in very early gestation raises the possibility that perhaps these cases of idiopathic preterm labor might be due to something that's happening much earlier than we currently are thinking, potentially around the time of implantation or even prior to that.
So you and your team have told a beautiful story of how histone modifications set early in pregnancy can shape labor timing weeks later. Like any great discovery, it raises even more questions. So what's next? What directions are you excited to explore moving forward?
So I think actually there's three big things that we're thinking about right now. One is we have this timer that's epigenetic in nature. And one question is, what genes exactly are this timer controlling to actually set the gears in motion at the end of pregnancy to transform the myometrium into this contractile tissue? Second question is, what's actually controlling the timer, both winding it up and programming it in early gestation, and what's controlling its erosion in the rest of gestation? What are the actual proteins that are involved with that, the epigenetic modifiers. And then the third thing that we're really interested in is to understand how this timer intersects with inflammation-induced preterm labor. So, as I mentioned, you can have an infection or inflammation that can cause preterm labor, and that seems to be intersecting with the timer in ways we don't really understand right now.
Super exciting future directions. That's all we have for today. So thanks again for joining us to share a little bit more about your cool science, and looking forward to seeing you all next time.